Welcome to Executive Insights for Life Sciences Innovators, and thanks for joining us today. On this podcast, we'll explore leadership, innovation, and growth in the life sciences sector. I'm Don Alexander, founder of GeneCoda, a boutique executive search firm dedicated to connecting visionary leaders with life sciences companies, shaping the future of therapeutics and scientific tools. On each episode, we'll dive into candid conversations with industry pioneers, uncovering strategies, insights, and real-world experiences that drive success in our industry. If you're interested in presenting as a guest of future episode, please scan the barcode image at the top of my video frame and follow the instructions. Now I'm pleased to be joined by Michael Heltzen, the CEO of eXoZymes. Welcome, Michael.
Thank you very much, Don.
Excellent. So Michael is the CEO again of eXoZymes, NASDAQ ticker eXoZ, a biotech platform company enabling the next generation biomanufacturing. Making very valuable natural product-inspired molecules, which are otherwise impossible to manufacture. Michael is originally from Denmark. He's a serial entrepreneur who's led multiple leadership roles in startups and their exits, including CLC Bio to Kyogen and Cardia Bio to Paragraph. Michael is passionate about the cross space in between computational power and molecular biology, and is dedicated to building companies based on good values, trust, and human growth. So, Michael, again, thanks for joining us. Let's talk a little bit about your career journey and leadership
Sure.
background. So, you've worked across entrepreneurship, strategy, and company building. What drew you to the idea that biology, treated as a technology stack, could become a major business frontier?
Yeah, that's been one of the cornerstones in what we have been building on. You're clearly talk to my team because a lot of the introduction here is kind of who I am, how I see the world. So I I see myself as a translator of very large technology breakthroughs into how they are going to make impact on the world. And that is the commercialization journey of figuring the science out, not just for the scientific science reasons, but for the like, how do we, instead of it can also do this, it can also do this, how do we take the smart choices on what it's going to do first? In which order are we going to build things? How do we focus and strategically align with whatever megatrends are in the world and the markets? And how do we do all of those things? Very important with the technology, very important with the market fit and so on. But third bucket is the team. How do we set a group of people up for success so that they, as individuals, especially as a group of people, become a high performance setup that can deliver on what is sometimes, because I'm a visionary by personality, goals that are maybe when the first time the team sees it, they go like, but that's 10 times higher than it's possible. And then it turns out that that it's not.
Got
So that's my role, and that often translates in. I have translated into to either and a co-founder role and a CEO position in the startup and scale up years.
Absolutely. That's great, Michael. When you stepped into the CEO role, what convinced you that eXoZymes was, I guess, what I'm going to call more than a clever scientific concept and had the makings of a real platform company?
So it initiated because of MDB Capital that I knew from my former life in the last company. We had basically a board member of MDB was an investor of mine in the former company, and then I was tapped right after we had sold the other company and said like, hey, we have actually this really big breakthrough over here, but we're struggling with like how do we translate that into commercial relevant things and what should we focus on, and basically a good fit. But then they said we're going to run enzymatic pathways outside of the cell as if it was chemistry, and then I said BS. That's not possible. So then I also thought like, but the guys over at MDB Capital, they're really smart dudes. So how they've gotten themselves kind of conned into believing in this, and people that knows Chris and the team at MDB, they're pretty convincing guys when they have done their diligence on something. They can answer any question you can come up with. So there was clearly something behind it. And then, to be completely honest, it was like a simple visit meeting the two technical co-founders, where it's just so clear that despite we're talking about something that goes against everything I had learned about biology, then it was working. It was working in the fundamentals, and it was working in a couple of applications that I could start foreseeing. Okay, if it works in that, then we could probably build something that's even bigger. After that initial meeting, my entrepreneurial mind didn't stop. I have a mind that is never turned off, basically, and I don't really always control where the thoughts go. I'm more of a viewer than the orchestrator sometimes. So my mind just started building with this new generation of technologies, and I relatively fast realized that they had made a tech breakthrough that we can talk about a little bit more here later on how biochemistry works. That basically that it doesn't have to be either you harvest something from plants or you do it with petrochemistry. There's a third generation. And people have heard it before, this value proposition via synthetic biology, but I had looked at that so many times and understood why that doesn't scale, why it doesn't get commercial business, and in my book doesn't make real impact on the world. Therefore, it didn't have my interest as a seed investor or as a participant in building those kind of companies. But they had solved a lot of, and honestly, all of the fundamental problems with SynBio, and therefore all of a sudden it clicked for me, and I was just like, okay, we're talking about a new generation of how humankind can do chemistry, how we can build matter. And then my mind also started saying, but these guys are not using AI to the degree I would have done, because I come from a line of companies where we used machine learning and AI methodologies to benefit from the different things we were working on. And it was just obvious to me that here we were generating some very large data sets with some very interesting patterns. Um, pattern discovery. Pattern discovery is what people should call AI and machine learning. It is just patterns in data. The same way our experiences and what we learn over time is patterns that we store in a very interesting way via our brain, but here via data that lives on computers. And the ability to sit down and have good conversations with the team at eXoZymes and talk about like how do we want to build this company? How do we also, frankly, how do we cut down our focus? Because the idea of focusing on 5 different things is not focus. So how do we get the strategic focus? What should the executional plan be? Was basically a 3 to 6 months period where I was working as chief strategy officer before I took over as CEO.
Yeah, no, that's great. I'd love to explore a little bit more on detail on that. Let me ask you this though first. How would you, I guess, characterize your leadership style as eXoZymes is trying to balance its scientific ambition with public company discipline?
You had a description written by my team that I actually think hit pretty precise, hit close to home for sure. I have a relatively different leadership style from the perspective of um, I am hyper focused on setting a team up for success in flat organizations. So I delegate a lot of responsibility out in the organization. And why do I dare to do that? It's because I know every single person on my team have been not just trained, but have adopted our 8 leadership principles. So basically, we have defined leadership principles in the core ways of taking choices, the core ways of behaving, the core ways of basically how we conduct ourselves as individuals, as a team, and as a company. And it means, frankly, now when things are set up and running, I'm not that needed other than I am then for the next layer of the puzzle. But my job, I try to think of as like how do I make myself not needed? How do I empower the organization to be smart enough as a group and motivated enough as a group that there's absolute no need for micromanaging. If anything, actually, if I need to get myself involved, it's to get people to go home from work instead of showing up at work. It's that's a positive problem, obviously. Making sure people remember that it's a marathon and not a sprint we're on. And it is a very exciting time because when you get these positive effects of, for example, paying forward, actively trying to set other people in your organization up for success because you know everybody else is going to do that for you, and you don't have to be afraid of politics or that there could all of a sudden be a decision taken that basically would fall down on you because the leadership principles doesn't allow for that kind of behavior. Then people dares to start taking chances, and we talk about we don't fail, we just fall, and that's a way of knowing that we were pushing the limit a little bit too much. But if we don't fall, sometimes we're not pushing hard enough. And it's also, frankly, an opportunity you give to your colleagues to save you, to catch you. So don't be afraid of kind of like really leaning in. And the eight leadership principles are in rough overview, heavily inspired by the Seven Habits of Highly Effective People, the book 30 years ago. That is basically a personal development growth kind of book that I believe there's been some editor at some point renaming the book into what it's then ended up being called, but it should have been called the seven and now eight principles of balanced living, because that is what it is. It's basically principles of like how do you find direction in life? What are your motivational factors? All of those really, really fundamental things. Those are the first, basically, three leadership principles. And the next three principles is about like how do you interact with other people successfully? And some of it is pretty darn simple. It's kind of like seem to understand before you push to be understood. It's basically learn to listen. It's surprising that we live on planet Earth. We are so evolved intellectually, and I would argue that more than 50% of people have still a lot to learn when it comes to listening. And I'm being very, very generous with the statistics here. I'm trying not to insult too many people, but honestly, maybe we should just introduce it as a course at university. Understand?
Yes,
And
that's good. That's good, Michael. Stephen Covey is one of my favorite. That may be one of my favorite top five, certainly top five of all time. So,
yep, I agree.
for those that haven't read it and digested it, they definitely should.
And it's basically a book that have already been translated into like the seven habits of highly effective families, the seven habits of highly effective, basically fill in your blank. And I've just done a practical translation into and the reason why it's like 7, 8, that's they kind of realized an eighth principle after the book was set up. It kind of makes it a full loop. We call it teach and inspire. So the principle is basically before you really have learned something, or you haven't really learned and understood something fully until you have taught someone else. Because what happens is when you take information in, you kind of make it fit into your reality. Now you can remember it, now you can believe in it, and you can use it for sure. So it's the biggest part of the way. But until you have sat down and translated that into words and convincing words for someone else so it fits into their reality. What you haven't done is like you haven't fully bought into the concept. So it's actually very self-motivating to teach other people things. So things you know already are much, much easier to exercise if you teach them to others as well. And frankly, it's also down to like, I can't really teach the guys over here how to behave and then misbehave myself and expect any of this to work out. So you keep notching the bar up on yourself as well.
Absolutely. eXoZymes sounds like it has a very empowering environment as a cultural norm. Let's switch gears and talk a little bit about the technology, what the company's developing. For listeners who don't live and breathe enzyme engineering, what is the practical difference between a traditional cell-based approach and the cell-free model that eXoZymes is building?
Yep. Because you and your audience is uh, already very knowledgeable, I'll kind of gear shift up my way of talking about it a little bit more. But if there's a layman or woman listening into the conversation, then we do have a number of podcasts and videos on our webpage that takes the language down. But basically, I would love to dive into the nuance of biomanufacturing and be a little nerdy about it. So let's just set up biomanufacturing in general. It's this idea of using biology to make chemistry. And we have biochemistry inside of plants and all kind of other cells. And what we basically with synthetic biology have been postulating and dreaming about is like, can we take those cells and make them into chemical factories for us? Because I have this cell, it makes a little bit of this compound that I want. Can I basically tweak the genes so that it makes even more of it, and then I'll harvest it out, and hence I have a business. This vision of cells as chemical factories forgot to talk with the cell about what the cell wants. Cells fundamentally are set up with 3 billion years of evolutionary pressure on only making chemicals it needs and only in the amounts it needs it. Everything more than that is destructive for survival. So you have not just a few, but a lot of pushback mechanisms that will downregulate if you, for example, introduce a gene that starts making a chemical the cell doesn't need or in amounts it doesn't need it. And the cell will fight back as if its life depends on it, because it literally does. You are polluting the cell with a chemical that is toxic for it and will most likely, in most of the cell-based setups, kill the cell in the end because you force it to do it. And just to continue this picture of the cell being a chemical factory, imagine that it was a factory. Then not only is it a factory that doesn't want to produce what we want it to produce, that burns down when you turn on the production. Number three, and that's where the business case really is killed, because you get your product embedded into the cell and mixed up with a million other things that is going on in a cell, your isolation cost often ends up being higher than the value of the compound you're producing. That means you have no business case no matter what you do. And that's basically how Synbio died on the vine. And that was where Paul and Tyler and Jim at UCLA sat a decade ago, 12 plus years ago, sat at UCLA that just published a cell-based biomanufacturing system that had been published in Science, and they were celebrating. And then, at least the way I tell the story after I've puzzle piece things together, because I wasn't there, but basically Paul and Tyler said like, hey, great publication, great fame we got out of that. But this is never going to work for real. This is never going to scale up. And we want to make real impact on the world. So that's a problem. And then the professor, halfway kidding, halfway in frustration, said like, okay, then just give me the enzymatic pathway. That is the only thing we need without the cell. And he basically went out of the lab with that one. And they were, as I remember Tyler describing it, well educated but young and naive enough to say like, yeah, why is it we don't just walk on water? What is it that is actually holding us back from running the wonders of enzymes without the support of the cell? And just for the few people that doesn't know enzymes too well, you basically have a feedstock or something. Think of it as, for example, a sugar molecule. You have breakdown enzymes that one step at a time breaks parts of that molecule off. That is the building blocks that the built-up enzymes then ends up assembling in 3D back to chemical 3D back into whatever small molecule we're talking about. So that's the enzymatic pathway, the breakdown and the build-up. And they basically said, like, what is it that a cell is doing for an enzymatic pathway? And other than half of that is our trade secret, and therefore hard for me to talk about here on a live podcast. I can basically talk about the things that we have published on and we have patents on. So basically, it's things like how do you ensure cofactor generation? How do you ensure that thermodynamics are working and the process doesn't run amok? How do we make sure the right building blocks is present that just naturally is there by the cell because it's a part of the whole cell universe anyway? So that took quite a while for them to kind of figure that one out. So that was an interesting academic journey. But in 2019, they then teamed up with MDB Capital as a co-founder of the company from the business side and financial side, and the scientists from the IP and science and entrepreneurial side, and they form the company that is today eXoZymes.
Absolutely, Michael. That's a great overview. Thank you for that. You touched on this earlier. I wanted to ask you a little bit more about it. So the platform itself spans wellness, pharma, and industrial applications, as I understand it. How do you decide where to focus first so the story doesn't become too broad to really execute upon?
Yeah, exactly. So you're exactly right. What I am saying, and it's true, this is a new generation of how to do chemistry. It's literally everything chemistry that you could try to build a cell-free solution on. And that is running off in all directions at the same time and complete lack of focus, boiling the ocean kind of challenge. So my job was to sit down and figure out where's the best product market fit or technology market fit, and how do we get ourselves into a position where the flywheel of creating an innovation, productizing an innovation, commercializing that product, getting that revenue back to the core source again, so we can run that over and over again. Because obviously this can be applied for honestly thousands of things, but like, how do we get started? And my thought process was a couple of different things, because we were working and had grants on a number of different areas in the nutraceutical pharmaceutical space. We had in biofuels, we had in food colors and different things. And it's like, how does that all fit together? And what was obvious to me was like, let's go after the low hanging fruits from a perspective of it has to create a lot of value, but it also has to be relatively quick in getting us there. And with the biofuels, that we still have a program on, and we are very proud to be partners with the DOE in pushing that forward. It is nevertheless a little bit on the back burner, going slow, because the reality is like we will only have sustainable aviation fuel when a lot of things have happened in the world, and they're taking their time. Let's say it like that. Where that was also because when I then looked at it from a financial point of view, a gallon of sustainable aviation fuel will be able to be sold for like $9. When I was talking with the pharmaceutical partners, a kilo, much less than a gallon, could be sold for hundreds of thousands to maybe $1 million. I was just like, why are we messing around with something where we need to put hundreds of millions of dollars of steel in the ground to get up to a volume where when you start delivering super tanker after super tanker of sustainable aviation fuel, it will be a great business. But I am, as the entrepreneurial CEO, given the task of booting this up in a lean fashion. So how do we get to value creation as fast as possible? So that led me in the direction of pharmaceuticals. Then at the same time, it started clicking for me, because I was like, hey, hold on. the nutraceutical business side, that is basically natural products as you can find it out in nature, but there's not enough of it. You end up stripping off all the bark of a certain kind of tree, and the trees die, and you won't have any supply chain after a year if you go that route. How do you find that nutraceutical? That's just one example of many examples. How do you basically get into that market that is also high valuable compounds, but at the same time, the route to market is much easier from a regulatory point of view. And that's where the double dip strategy that we call it is like, okay, we will focus on very valuable nutraceuticals that we can build a biosolution for and get to market with fast. And it also has to live up to that we can then take that same molecule because of how the platform works. We can not just make the natural product version, we can now start making new to nature versions that are much more potent or more bioavailable. We basically take advantage of this core platform that is a new generation of chemistry. So we can expand the white space of small molecules significantly. And that's where the pharma companies are waking up and saying like, oh, that's really interesting because we have been working with this natural product and it did do a little bit of what we wanted to do, but it wasn't potent enough, or it was like fill in your what the problem was, why it wasn't commercialized. So we basically took that as our strategy and then started building the search parameters of what the best fit would be. And it's basically down to have something already been realized to be very valuable. We love when other people have spent $100 million on mapping out for the world why this compound is super valuable if they only had something. So this is why it didn't progress through the clinical trials or something like that. And we can look at that and go like, we can uniquely make that. Not only are we the only people in the world that can make this compound, but we can also get a composition of matter patent on new versions because it's new composition of matter.
Yeah.
And that's the pharma game. So that's kind of how we got it all narrowed down to nutraceuticals with a pharmaceutical use case. And we have now picked our two first lead programs and are making those products progress in basically building the biosolutions and bringing it to market. We can talk a little bit about those products, or we can talk about other things as well.
Oh, no, that's great. Really appreciate that overview. It seems like a very well thought out and planned strategy. I guess one question is around the toughest challenges in taking eXoZymes from earlier in its scientific kind of discoveries promise state to more of a commercial readiness application.
Yeah, we're not the only company in the world that have lifted an enzyme from inside of the cell, outside of the cell. That scientific journey of like genetically optimizing the enzyme to be able to do whatever you want it to do. So some things it was literally just getting it out of the cell and then it was working. So if you look at some of the enzymes that are in laundry detergent that breaks down stains, then they're relatively close to their natural cousins. So we benefit as a company from pioneers that already back in the 80s and much before that were working on like how do you get an enzyme to work outside of the cell and how do you kind of do that? Where we're unique is like we then took it to the extreme and said like we don't want one chemical reaction only. On the whole enzymatic pathways. We build pathways all the way up to 30 enzymatic steps from feedstock to end product. This is where people go like, okay, this is not possible. This must be bullshit. Exactly as I said it. So we still have some of that education. The good news is that it's up and running, so people can just come and visit us and kick the tires on it. It's not everything we can explain how we got to work, but it works in front of people and it's real. So back to your core question, what really was a bottleneck when I came into the company, I started as CEO in 2024, was we could evolve the enzymes to become eXoZymes. That by our definition is basically if enzyme is thought of as like inside reaction, then eXoZyme is the outside reaction version. We sometimes call it the superhero version of the enzyme is an eXoZyme because we want want him, if he's a little worker, to basically be in a much harsher environment that he's used to, only do one thing, do it really well, and do it under the conditions we decide that he needs to live under. And we genetically want to push him to evolve in that direction. So out in nature, natural evolution is like one lifetime per mutation. If that leads to a better trait or feature, then that becomes the prevalent trait, and that's basically how evolution have gone over the billions of years. We basically sit down and we brute force evolution. We sit down and calculate what is the mutation that potentially could lead to an improvement, and we try to come up with all the things we can calculate that potentially could lead to an improvement. And then where there was a huge bottleneck was people were saying like, yeah, that's good, Michael. Now we are per enzyme projecting 1,000 optimizations. That's just going to take us 100 years in the lab to test out all of those by taking that DNA and putting it into a cell, growing it up, harvesting that, harvesting that enzyme, testing that enzyme, and then seeing if we were right, and then do that 1,000 times. We see each other again in 100 years. And I was like, nope, we need to find a way where we can minimum 100x our throughput. And the team first a little, okay, that's a high bar, but as it happens when you have really strong culture where people dare take chances, there was finally someone that said like, you know what, I have an idea. Maybe we could try this out. We are experts in cell-free environments. Why don't we build a cell-free environment where we put DNA in, and then there's all the enzymes and small things needed to express DNA into enzymes. So why don't we do cell-free protein or enzyme synthesis? Why don't we just express enzymes without use of living cells? And that's in itself a little crazy because anyone that have worked with cell-free protein synthesis, they know you get tiny amounts of the protein, and we need much more. And it's not just a protein; it's an enzyme that has a function. So prediction from the DNA sequence up to the protein structure is difficult. This is AlphaFold kind of methodologies I'm talking about. Then the next generations of going all the way to feature under certain circumstances. That is the computational side, and then this. Can we run 1,000 in parallel? But we can now. We can run 1,000 mutations in a week. We can run 1 billion years worth of evolution in a week.
That's
These
amazing.
obviously kind of just example numbers. I don't know what it actually translates into.
No, that's really amazing, Michael. What role, I guess, or how does AI add value to this equation?
Yeah, so it is machine learning algorithms that are so basically DeepMind in the UK that led the machine learning AI revolution of the world. That's now a part of Google and their Gemini team. The guys over there solved Go, the chess style Asian game first. And then after that, they were bold enough to say like if the patterns in language because it's large language models we're talking about neural networks. If the pattern language is so strong that we can do this with Go and what ended up being the language models, why shouldn't it also work of DNA to proteins? So this is the starting point of that thought direction. And we're probably on generation 3 or 4 now, and we have nerded ours out in the direction of enzymes specifically. And this whole training data set that we have built on like what does it take for an enzyme to work outside of a cell is obviously it's equally much the algorithms as it is the ability to generate the right data that teaches you the patterns. And let me just spell that out completely. So with 1,000 mutations, we go out, we express all of these different versions of the enzyme that we then take back and say, okay, this mutation, we were right about it. He now performs, or it now performs better. Our little eXoZyme friend, he now performs better with that mutation. So that's over in the pile of things we can build with. But equally interesting is like we predicted that this mutation would lead to an improvement that it didn't lead to. Now algorithms go back and learn from that. You were not We're not right about this. So this is called reinforcement training, where you get these circular loops. This is not something that had been available to biology and biotech before, because typically there's not a one-to-one correlation between what you change and up to the outcome, because you're living in a living cell environment. So you have like one mutation doesn't lead to necessarily one change only, because there's also a million other things going on, and it happened to be Tuesday instead of Wednesday when you ran the experiment, and therefore the experiment came out slightly different because it's living matter we're talking about. And that ability to start do reinforcement training is why we can brute force evolution, and why we can basically fast forward. And then we have a pile, in the end, we have a pile of mutations that will improve, and then we can start combining mutations. And that's how you get an exozyme.
That's great, Michael. A couple of last questions here. Looking out further, maybe 5, 10 years out, what kinds of molecules or product categories do you believe will best demonstrate the long-term potential of this system that you're building?
So we've obviously chosen our flagships to prove to ourselves and to the world that this new generation of biomanufacturing is here. So the first product line or program we have chosen is a small molecule called NCT. That's basically a small molecule that boosts metabolism. That means turning fat into energy. That's a good value proposition up against the backdrop of GLP-1s and the biggest biotech and pharma boom in history, the fastest growing, largest markets ever. And there is an opportunity with GLP-1s that is basically that's a large molecule that turns down your hunger signal, but it also turns down other activity signals such as muscle growth, bowel movement, other things you're gonna, at least some people are gonna suffer from if they take that also over a long time. And then it's super expensive. So then there also that element to it. So we have a natural product in NCT that if we could make it pure, we can basically get GRAS approval, generally recognized as safe by the FDA framework. And you have already had NCT. Everybody have already had NCT because it's in trace amounts in black pepper, for example. The only challenge is just like you will need to eat a houseful worth of black pepper to get enough of one dose of NCT. And that's just not practically possible and financially viable. So we basically did exactly that. We sat down, studied the genes behind the enzymes that does that trace amount, and then we ran this evolutionary optimization platform on it. We built an eXoZyme system for it, and we just recently basically showed our pilot publicly together with Cayman Chemicals that we could build a tech transfer package for NCT and hand it over, and just with kind of access to the building blocks and our mix of enzymes and the protocol they should follow, they could produce NCT at a purity never seen before, at a performance never seen before, and at a speed that is kind of outside of scale. And let me just put some numbers on that for the people that are in the industry. So the purity is 99.6%. The only other thing that is in the solution when it's done is a little bit of leftovers from either the feedstock or the intermediates while it's building it, and then the eXoZymes themselves. The rest is end product. We have a conversion of 99% of feedstock to end product. If you think about a cell that you give, let's say, sugar, and you have a pathway inside of the cell that makes your thing, if you get above 10%, you are really, really, really good. And there's few examples where it's maybe 50%, but like if you're not talking about the outliers, but cell-based in itself, like 99% is just, it's not comprehensible, because like of course the cell will use the feedstock for other things. Then there was the speed of it. If this was done in a cell line that made NCT, an engineered cell line, It's probably 10 to maybe even 20 times faster. And from a price point of view, the difference between running something for 4 days versus, I'm going to say 4 hours. I'm maybe a little bit unfair in my math here, but like we're doing it on the fly. Um, it's basically production cost. Like this, the equipment can produce at least 10 times as much with the same capital investment. That matters.
That's
And it
an
means
overview.
the pricing is because it's efficient and because it's fast, you can basically have profit margins that is unheard of otherwise.
Yeah, yeah, that's an order of magnitude for sure. There are other innovators, company founders that may be listening in today. I'm curious, you've touched on several already, but are there any other pieces of advice, best practices that you might offer to other founders or executives who are building platforms out there in today's market?
Yeah, my first advice I've given already is like, if you want to build a really deep technology company, focus on building the best possible team. I don't believe in the old mantra of like, we will just work people to their bones, because that's honestly not how innovation happens, at least in our industry. I'm not saying in other industries it can't be different, but if you want super creative people daring to take risk, then create an environment where that is enabled, allowed for, maybe even cherished, and set the feedback loop up so that you don't fail, but you just fall and learn from it. So that's the advice I gave already, and a lot of that way of thinking. Like Seven Habits is really like, if you have not read that book yet, you have done yourself a disfavor. It helps you understand how to live life as a person, how you interact with other humans, and a lot of other skills that I'm a very big fan of that book. So there's that, and then the last part is probably reminding people that feedback and criticism and all of that is just people's point of view. And if you have seen the light, the opportunity, and you understand how to get there, and you can break it down all the way from like, we will end up out there when the following things has happened, and these are the value inflection points. Don't focus on anything else. All the side quests, all the like, this could also be interesting over here. That is literally you not progressing. And every time you take a side step, the chance of you failing is significant. So focus is a hard learned lesson of my own that I wish someone would have taught me. And maybe it's a burnt fingers principle that nobody understands what it means that the water is hot until you know what it means. That boiling water is not something to put your finger into.
Fair enough, Michael. That's great. Well, that wraps up this episode of Executive Insights for Life Sciences Innovators. I appreciate you tuning in. Hope you found today's conversation valuable. If you enjoyed the discussion, please be sure to leave us a review and share it with your network. Please also mark your calendars for May 1st at 1 o'clock Eastern with Stan Wadowich of Ridgeline Therapeutics. Thanks for listening, and we'll see you soon.
Thank you.